So Zinc-finger nucleases, as I was saying, so we have a look at the history of the Zinc-finger nucleases from where they originated and their applications in today's modern world. Sorry, what are Zinc-finger nucleases? So, I mean, they are some kind of artificially engineered restriction endonuclease enzymes, which are generated to target specific DNA sequences within the whole genome. Specific DNA sequences of our interest, not those DNA sequences that are recognized by the normal type of restriction enzymes. But the question is, why zinc fingers, or what exactly are zinc fingers? So zinc finger, as many of you would be knowing, and of course for the uninitiated, they are domains of one of the most common structural motifs in eukaryotic cells. When you say common structural motifs, I basically mean that they are protein motifs formed from the secondary protein structure. And as you can see in the figure here, that it's made up of secondary structures like alpha helixes and beta sheets, and and they are very important proteins that are found in transcription factors, including TF3A, ZIF268, are to name a few. Now these DNA binding proteins, these zinc finger proteins, they contain up to 37 zinc finger domains, which are connected by flexible linker regions. So these are the linker regions which are connecting here. For example, the alpha helix is connected to the beta sheet, and this zinc ion, it is what is making this whole system or whole molecule compact, and and you can see that it is held from the zinc ion that is present there. Now cis2 his2, zinc finger proteins, that is cysteine 2 and histidine 2. That means two molecules of cysteine and two molecules of histidine. Zinc finger proteins are most of the common type of DNA binding domain found in the majority of eukaryotic genomes. So the motif, the peptide motif itself comprises of 30 amino acids that adopt a compact DNA binding structure chelating on a zinc ion. Now in the structure that is shown here, we can see three zinc fingers, which kinds of interdigitate between the major grooves of the DNA, or they are inserted in the major grooves of the DNA, and this is what the crystal structure of ZIF268 DNA binding domain shows. So the zinc finger array wraps around the DNA with the alpha helix of each finger buried in the major groove. Alpha helix of each finger, interdigitate in the major groove. So let's look at the structure once again. Cis2 his2 zinc finger protein. So you can see that these are antiparallel beta strands or beta sheets, and through a linker is connected to one alpha helix, and this kind of loop connects it to the other alpha helix. So there are the connectors can be loops, can be turns, and that's how they are linking from one alpha helix to the other. So cis2 his2 zinc-finger proteins. A little bit of history of zinc-fingers. You know, zinc-fingers were identified in a study of. It's not moving. Step in first. Share the screens. Yes. Share. Yes. This is true. This is yes. Step. Share. Yeah, now you can see. Is it changing? Yeah. Just check. Is it changing? Yes. Okay. So there was some kind of a technical glitch. I was told that the screen was not being shared or was not moving. So basically, we are talking of Zinc-finger nucleases, which can be defined as some kind of engineered endonucleases or artificially uh, engineered endonucleases that are generated to target specific sequences in the DNA, sequences of our interest in the whole genome. But why Zinc-fingers, or what exactly are Zinc-fingers? Are we seeing it now? Is it moving? Yes. Yeah, great. So as I was saying, Zinc-finger domains are one of the most common structural motifs in the eukaryotic cell. When I say structural motifs, I basically mean protein motifs, motifs formed from the secondary structures in protein, especially like alpha helix and beta sheets. And they utilize the motif in some of their most important proteins that are used in the protein transcription factors, like for example TF3A and or zinc or GIF268 are to name a few. Now these DNA binding proteins, they contain up to 37 zinc finger domains connected by flexible linker regions. So these are the flexible linker regions that connect the different domains or the different secondary structures. And these linkers are basically, these can be turns or loops, like for example helix turn helix, or a helix turn loop, helix turn beta sheet, or a helix loop beta sheet likewise. So cis2 his2, basically cysteine 2 and histidine 2 zinc finger proteins are one of the most common type of DNA binding domain found in the majority of eukaryotic genome. So the peptide motif, or this protein motif, it has about 30 amino acids, and which adopt a very compact DNA binding structure chelating on a zinc ion. Now this zinc ion, you can see in the figure the greenish, the spears, the greenish spears, which is holding the whole structure. There are a whole lot of interactions going on, which is held in place by these kind of zinc ions. So this is basically the crystal structure of ZIF268 DNA binding domain in complex with its DNA binding site. It shows how the zinc finger array wraps around the DNA, and the alpha helix of the zinc finger, it interdigitates into the major groove, or it buries itself into the major groove. That's where the interaction with the protein and the DNA occurs. So now let's look at the structure once again. Sorry, cis2, his2, zinc-finger proteins, and we see that these are anti-parallel beta sheets connected by the linker molecule, and then by another turn molecule, it is connected to a beta alpha helix, and then this alpha helix goes right up to the third alpha helix through turns and loops as the linkers. So history of zinc fingers. So they were first identified in a study of transcription in the African clawed frog, Genopus laevis. We will know more about it later in the laboratory of Nobel laureate Aaron Klug, and on their study of transcription of 5S ribosomal RNA revealed that the binding strength of this small transcription factor, this is TF3A, was due to the presence of these zinc ions, the zinc coordinating finger-like structures. So it was discovered in 1985. With the binding of transcription factor 3 with 5S RNA, and the subsequent crystallographic studies or structural studies revealed its three-dimensional structure and its interaction with DNA. So it's basically the zinc finger is a self-contained domain stabilized by this zinc ion, this reddish sphere that you can see, and it is, you can see that there are it interacts with two histidine molecules in the alpha helix. So there are two his2 and two cysteine molecules in the beta sheet. So cis2 his2. And it also has a hydrophobic core. It also has a hydrophobic core. However, the star of the research was the African clawed frog, Genopus laevis. So it was, you can see that the frog has claws. And of course, the other star was, Aaron Klug and his group. So the zinc finger discovery showed not only a new protein fold, but also a novel principle of DNA recognition. The other DNA binding proteins generally make use of twofold symmetry of the double helix, functioning as homo or heterodimers. But here you can see that it is basically very active in its three-dimensional fold. So these zinc fingers can be linked linearly in tandem to recognize nucleic acid sequences of varying lengths. So this offers, this modular 3D modular design offers a large number of combinatorial possibilities for the specific recognition of DNA or RNA, as the case may be. So it is not very surprising that zinc finger is found widespread in nature. Of the human genome, 3% of the genes have zinc finger associations. Now, so each zinc finger, the residues, they can recognize a triplet codon. So this finger 1 can recognize GCG, finger 2 can recognize GGT, and finger 3 recognizes GCG. It's basically, I just made it up so that you can understand the thing. But then there is another story to be told, the story of restriction endonuclease. So all of you, most of you are very much aware of restriction endonuclease, But for the uninitiated, restriction endonucleases are something which had revolutionized genetic engineering and biotechnology, and they are basically some enzymes which recognizes some sites in the DNA. And how did it originate? What is the story of restriction endonuclease? So bacteria has been found to be attacked by phages, that is bacteriophages. So these are, as you know, these are very small viruses which attack and which injects the bacterium with its DNA, and it kind of hijacks the bacterial protein production system or protein uh, synthesis. So instead of producing proteins that are required for the bacteria, now the bacteria starts producing, the proteins that is required for assembly of the phage virus. So this has been going on for, as far as evolution has been happening. And so you can see that the DNA is being injected, and as the cellular machinery of the bacteria has been hijacked by the viral DNA, so it starts producing the different parts of the viral DNA, and so these bacteriophages or phages are produced inside the bacterium, and lysis, by the process of lysis, it breaks open the cell wall, and they are released for further infection of new bacteria. But over millions and millions of years, the bacteria also have developed some kind of defense systems against such phage DNA. So we see that when the DNA is being inserted, the DNA is being injected inside the bacterium, there are a class of enzymes, as I said, restriction enzymes. They recognize the sites, in the specific sites in the viral DNA or the phage DNA, and they cleave it, they cut it up into, chunks, small fragments. So what are the chances of the restriction enzyme uh, uh, cutting its own bacterial DNA? Now that doesn't happen because the bacterial DNA is methylated. So the restriction enzyme can see. So methylation is a process by which a methyl group is attached to different regions of the DNA. It is almost like, when you go to conferences and you are made to wear conference, IDs or conference tags, which recognizes you as, that you are a part of the conference. So here also the same thing happens. So they are basically tagged. They are basically tagged by these kind of methyl molecules or CH3, and the restriction endonuclease, they recognize this spatial tags, so it knows what is me and what is not me, who is an outsider and who is me. So it will not destroy its own DNA. So this was the tug of war that was going on between the phage DNA and the bacterial defense system, so which we have used to our benefit. So Werner Haber, then Daniel Nathans, and Hamilton O. Smith. So they were the ones who had discovered restriction enzymes. So in 1960s, Haber, he observed a change in a bacteriophage DNA after it invaded resistant strains of the bacteria. So the fast DNA he found was degraded and cut into small pieces. So this was the first time discovery of restriction endonuclease, and for this they were awarded the Nobel Prize in 1978. Werner Arber, Hamilton Smith for the discovery of restriction endonuclease, and D. Nathans for demonstrating that restriction endonuclease can be used to physically map DNA. So restriction enzymes are traditionally classified into four types. Actually, it should be three types based on the subunit composition, the cleavage position of the restriction enzyme, and sequence specificity, and cofactor requirements. So the three categories of restriction enzymes are type 1, which recognizes specific DNA sequences, but they make their cut at seemingly random sites that can be as far as 1000 base pairs away from the recognition site. For example, in ECO-B and ECO-K. Sorry. Type 2 recognize and cut directly within the recognition site. Example, Hindi 2 and Hindi 3. Type 3 recognizes specific sequences. They make their cuts at a different specific locations, which is usually within 25 base pairs of the recognition site. So the first restriction endonuclease characterized was isolated from the bacteria Haemophilus influenzae. So the enzyme HIND2, it is written as Hind, but it's mostly pronounced as Hindi. Hindi 2 cuts at a particular site within a specific sequence of 6 base pairs as follows. So you can see the recognition site for Hindi and the cleavage site, which you can see here, how it cuts. So of course the nucleotides, you can see they are shown in parenthesis, and the enzyme, it recognizes them. So EcoR1 is another enzyme which was isolated from Escherichia coli. It recognizes and cleaves a palindromic DNA sequence in a staggered fashion to form the sticky ends, which you can see here. These are the sticky ends. Okay. So we now have generated a huge catalog of bacterial restriction enzymes that were gradually discovered. You know, that the different crystal structures have been worked out since 1970s, and how they recognizes specific palindromic sequences. So palindromes, as you'd be knowing, these are, you can see in the 5' 3', this is red is GAATC. Now when you look at the complementary strand from the other end of the antiparallel strand, it also reads GAATC. So most of these sequences are palindromic in nature, and they are recognized by the restriction enzymes. So this huge catalog shows us that there are about 3,000 restriction enzymes recognizing over 230 different DNA sequences. And although they are mostly found in bacteria, but they have also been isolated from viruses, archaea, and eukaryotes. However, there is a problem with restriction endonuclease. And the problems are that they are very recalcitrant, and you cannot modify them. You cannot persuade them to cut sequences other than their sequences that they can recognize. You cannot modify them to cut other sequences. Even if you can, the signal of such cutting, or they cut it very weakly. So this question Yes, I mean, whether such enzymes could be modified to cut desired sequences. Uh, Not exactly. And why? As I told you, the the primary reason was that most restriction enzymes, they recognize specific sequences, those palindromic sequences, if you may. And the second reason is that they recognize very short sequences. Now, when you recognize very short sequences, there are chances of that short sequences to occur many times in the genome, and they will thus cut the genome many, many times. So, as I said, so these restriction enzymes cannot be modified or coaxed to cut other sequences. Say it cuts the sequence X, you cannot modify it to cut sequence Y. Even if they are persuaded to cut such sequences, they cut it very weakly and leading to such uncontrolled cuts which are slightly off the mark. Sometimes it is way off the mark. So this definitely was not the route to gene or genome editing. But then we were saved by type 2S or shifted cleavage type of restriction enzymes. So they are partitioned into separate domains linked by a short polypeptide connector. As a result, type 2S are larger proteins than type 2P, and they are typically 400 to 600 amino acids in length. So when these type 2S enzymes bind to the DNA, the catalytic domain is positioned to one side and several base pairs away from the sequence bound by recognition. So as I said, there are two domains. One is the recognition domain and one is the cleavage domain, the catalytic domain, which actually cuts the DNA. But it doesn't cut the DNA where the sequences are being recognized. It can cut away. And what sequences, it doesn't matter actually. It can cut any sequences. So that was very much to our benefit. So you can see type 2, normal type 2, it cuts the DNA at the place where it is bound. Whereas in this shifted cleavage, you can see that it is recognized in this particular, say, GG, TT, TC, TC, and CCA, GAG. It is not cutting at that point, but several bases away at this, which is the cutting site. NN, basically denoting the cutting site. The same thing here below. So these type 2 enzymes, they generally bind to the DNA as monomers, and they recognize asymmetric DNA sequences which are not symmetrical. So they cleave outside of this sequence within 1 or 2 turns of the DNA. So, say around 20 base pairs away, they cut. By convention, the recognition sequence is written in orientation in which the cleavage occurs downstream to the right of the sequence. So as I told you, the cleavage produces staggered ends of 2 or 4 bases. The exact position of the cleavage are indicated by number of bases away from the recognition sequence in each strand, both the coding strand and of course the template strand. Now, this we will go to the next slide. So however, we were saved by a game changer. So this game changer is a type of restriction 2S, and it is the FOC1 enzyme. So it is the classical example of restriction enzyme. Restriction enzyme 2S, and this is the restriction endonuclease FOC1, which is naturally found in the bacteria, Flavobacterium oquinoans. It is a bacterial type 2S restriction enzyme consisting of N-terminal DNA binding domain and a non-specific, the cutting or cleavage domain at the C-terminal. So FOC1, the FOC1 enzyme, it recognizes non-palindromic sequences, in the duplex DNA. And the recognition domain of these proteins are helix-turned helix domains. So they basically cleave at 9 base pairs and 13 away from 5' and 3' respectively. So now this figure or this cartoon shows the dimer molecule. Of course, this FOC1 there are two molecules. You can see the green and the blue ones, the light blue ones. However, each molecule has two domains. So you can see this in the bigger one. This is the recognition domain, DNA recognition domain. So it will come and bind somewhere here, and this is the cleavage domain. Okay, so it will go and cut somewhere here away from the recognition domain. So these are some more details. I will not go into this. We'll go into the basically how the FOC1 dimer works, how the molecule works as a dimer. So this gentleman that is shown here, he is one of the pioneers. He is one of the main players who had fused the FOC1 cleavage domain with different type of Zinc-fingers which recognizes some specific sequence. So in 1996, three scientists, namely Y.G. Kim, J. Cha, and S. Chandrasekaran, they reported the deliberate creation of novel site-specific endonucleases by linking two different Zinc-finger proteins to the cleavage domain of FOC1 endonuclease. And this was for the first time they were able to create artificial nucleases that will cut DNA near a predetermined site, a site that is to our interest. So now it was possible to have or to design programmable nucleases. So where the FOC1 catalytic domain can be fused to different DNA recognition domains, which are basically your Zinc-fingers. So the Zinc-fingers are most common DNA binding domains in mammals, as I had earlier told you. So almost 3% of the human genome and more than almost 1500 human genes has Zinc-finger motifs, and 720 Zinc-finger family genes encodes basically the same cis2 his2 Zinc-fingers. And so basically this is basically a beta alpha fold, and of course, as they were discovered in 1985 and they were studied in the 1990s, and a gradual emergence of the DNA recognition codes from the phase display experiments. And so this knowledge that was being gathered, it made them it made them make this kind of arrays, this kind of Zinc-finger arrays, which will cut the DNA at our site of interest, in the sites of interest. So this is the FOC1 enzyme. You can see we are only using half of the dimer. This is a monomer which is used here, and the other monomer comes in the complementary strand. What is the exact mechanisms? We will go later. We will go later. So as I mentioned, that FOC1 cleavage domain has no specific sequence or sequence specificity, and basically functions as a dimer, as I told you here. And there are about 6 nucleotide bases that it covers. So 2 zinc fingers needs to be made, and this increases the sequence specificity. So one is this and the other is this. So in each of the assembly, in each of the array, you can see that there are about 3 to 4 zinc fingers. So these are your zinc fingers, and these are all different zinc fingers which recognizes the bases. So 2 to 3 or up to 2 to 4 zinc fingers. Mostly they are 3, sometimes it's 4, and they are attached to the fork 1 enzyme. So this is your fork 1 enzyme, these green ones. And so ultimately we have 4 fork 1 enzymes. So they will cut the DNA at 2 specific sites. And these whole intervening sequences would be excised out. How it does, we will gradually, I will show you it in a step-by-step process in one of the subsequent slides. So zinc finger nucleotide nuclease base genome edit. So this is your left zinc finger proteins, and this is your right zinc finger proteins. So the left is at the template strand, and the right is on the coding strand, and so they will now they will dimerize the fork ones, which were half the fork ones that we were using. They will dimerize and they will cut the DNA at that particular site. You can see here the old stranded breaks here. So so this can be done by homologous recombination or non-homologous recombination. Where in homologous recombination, you can see that the gene correction can be done by DNA insertion, or in non-homologous, and it can cause gene disruption. So basically, so these bold structures that is shown here are the structures that has been cut by this kind of mutation, this kind of zinc finger nuclease mediated cuts or excisions, and this will be excised out. And of course, this is not a frame shift mutation. Why? Because the triplet codons, or the 3 plus 3, that is 6 nucleotides, would be excised. So there is no question of frame shift mutation. Now, a foreign DNA or a DNA of interest, which can be generated in the the synthesizer, can be put in there, and we have our new gene which has been edited, which is markedly different from the existing gene or existing sequence. So the zinc fingers, basically, as I told you, you can see the different types of amino acids, leading to your different types of recognition. So so this, for example, arginine 6, lysine 6, then serine, phenylalanine, they recognize, all of these recognize G, the nucleotide G, whereas glycine 6, it recognizes A, then serine recognizes C, then lysine 6 and aspartate, they recognize T. So ultimately, one zinc finger protein will recognize 3 DNA sequence, which is, you can say that it recognizes a triplet codon. And so these zinc fingers, this particular zinc fingers, you can see it is wrapped around the DNA. And of course, this is your cis2, his2 zinc fingers, which will help in cutting, recognizing and cutting the DNA at the particular sites of our interest. So let us look at the mechanism of a Zinc-finger nuclease genome editing. What is this mechanism in a step-by-step manner? So one Zinc-finger, this will basically recognize three bases in one side, and then another Zinc-finger will come and bind, which will recognize another base pair, another three such bases. So two Zinc-finger, 6 DNA bases. So basically 2 amino acids. Then 3 Zinc-fingers, they will recognize 9 DNA bases, hence 3 amino acids. So this is more specific target site recognition. So overall, there is 9 to 18 base pairs that are recognized, and 3 to 6 amino acids are recognized. So this is one side, it's happening on one side. So now these Zinc-finger are fused with the DNA nuclease domain, that is half the fork-1 endonuclease. So this is just the half of fork-1 endonuclease. As you see, the fork-1 is a dimer. So we are just adding the half of that dimer. And they cannot cleave, they cannot cut until and unless they are dimerized. So we are providing them the other array of Zinc-finger fused with fork-1 enzyme, and which is the other monomer or other part of the dimer. So now this two half of the fork-1 have have dimerized, and when they dimerize, they have cleaved the DNA at this non-specific site, or rather the site of our own specification. So as a result, you can see if we add, say this was one such or two such arrays. Okay, let's go back. So this array here shown here is 1, and there is another one here. So we have 2 cleavage sites. So we need to cleave it at 2 portions. Okay, so that we can remove or we can excise this intervening DNA sequence. So ultimately we have such 4 Zinc-finger nucleases, that can cleave at 2 different sites, so that the intervening sequence can be excised. So what happens after this event? What happens after this excision is carried out? So this intervening sequence, the DNA fragment which is lying between the cut site, is excised, it's thrown away. And a new synthesized strand may be inserted, or it could be foreign DNA containing homologous ends can also be added. And this homologous recombination, which I had told you, occurs leading to the insertion of the new foreign DNA or the newly synthesized DNA. So now that we have understood, I believe you have understood the mechanism. It is time for us to look at the application of Zinc-finger nucleases. A few case studies. So Zinc-finger based HIV gene therapy. Now let me give you a brief background. Most of you would be knowing, but then for the beginners, so I would like to recall that HIV recognizes CD4 T cells. These CD4 T cells are basically, lymphocytes, T cell lymphocytes, which recognize, which has a, receptor at the outward wall, which is called CD4 receptors. So what are CD4 receptors? CD4, basically CD stands for cluster determinants. These are receptors that are recognized by HIV. It is almost like a doorway by which the HIV can enter into the these helper subsets of T cells, that is CD4 T cells. So it acts as a doorway which allows the CD4 cells, or which allows the HIV cells to enter into the T cell lymphocytes, this particular type of T cell lymphocytes. But it also needs a cofactor, CCR5. So what is the function of this CCR5? It's almost like a gate man. It's almost like a, Darwan standing at the door who ushers you inside. So this CCR5, this cofactor, ushers or helps the bacteria to get inside the CD4 cells. And when it's inside the CD4 cells, it is very difficult to destroy this bacteria by, different retroviral medicines, which combats the bacterial, the viral propagation. So there were a lot of ideas generated in the past where they were thinking of knocking out this CCR5, which is the cofactor. But it was not very successful. They tried with different methods of knocking out. They tried with antisense RNA and RNA silencing, but it was not knocking out. It was not working out. But these kind of nucleases or designer nucleases, like for example Zinc-finger nucleases, could very easily knock out the CCR5, which actually stops the entry of the virus into the CD4 cells. So apart from that, Zinc-fingers were created to, the first ever targeted knockout rats. So why do you need knockouts? So that we can study the different pathways, So you knock out a particular part, a particular, relay of a knockout, a relay of the pathway, and you see what kind of perturbation happens. So there were a lot of success with such, Zinc-finger nucleases, which were able to knock out different types of genes in the rats. So they were able to come up with a knockout rat model, by using such kind of knockout systems by Zinc-finger. And so micro injection of Zinc-finger mRNA and donor plasmids, they were added to single rat embryos. I will of course not go into the details. This can be discussed in some other topic. So so then you check the changes in the target gene expression using quantitative PCR. So these are the changes that can be brought out, and I will again not go into the details. And then we come to see what are the problems with the restriction, Zinc-finger domains or Zinc-finger nucleases. They basically, you can have good gene therapy or good gene editing therapy, and it is easy to manipulate. And the best thing about it is that you can target any site in the genome. But of course, when you look at the specificity, it may sometimes go off the target. So there are many cases where it is not exactly in the place where it was intended. And of course, then talents came. Talents is another such nuclease mediated cutting system or cleaving system, which was more efficient, found to be more efficient than Zinc-finger. Of course, you'd be knowing about talents in the subsequent lectures. Then another important thing is, of course, the ethical and legal implications. You know, like any medical treatment, the consent of the patient is required for a potential zinc-finger therapy. Not only that, and there has to be huge amount of data, about the trials in different systems, different animal or other such systems, and only then you can, you will be given permission by a regulatory authority to try out in human trials. So engineered nucleases like ZFN can often have off-target effects. So we are not very sure whether it's going to, be 100% cure or not. And the use of embryos to edit genes, to create superhumans or gene-edited super babies is something we should be considered with some amount of caution. Hello. So with this, we come to today's the end of today's lecture, and if you have any queries or questions, I would be happy to answer them. Share. How can I see it? Chat box. Yeah. Yeah. This one? Hello. Yes. Yes. Stop sharing. Huh? Everybody. Chat box. Never. What's your calendar? This. Which one? Slide is not working. It was one on the video. I will go. Okay. Yes, we can call them genetic seizures. And what type of ailment could be treated by zinc-finger therapy? The possibility is huge, but of course it has to be more specific. You cannot have off targets. Suppose you target one particular sequence and it goes and cuts, say 3 or 4 base pairs away, it's going to create a lot of problems. So although it was used in checking the entry of HIV, and into the CD4 cells, and also it was used for developing rat knockout systems. But we, as I said, we have to maintain a lot of caution. We don't know what it can do. But of course, before it can be widespread, talents came up. After talents, even if before talents could be become more widespread, you know, CRISPR-Cas9 came up as a new, and it is one of the latest of gene editing system. Can we remove any genetic disorders during the lifetime of an individual or only during embryonic stage? Now, there are two things. One is when when you use it in the embryonic stage, so it can be found in almost all the somatic cells, and there is a possibility that it can go for vertical transmission. That means from father to children or mother to children, it can go. It can go, vertically. But when you correct a few tissues in your somatic cells, this will not be uh, vertically transmitted. And we can, of course, our whole the whole idea is to correct this kind of genetic disorders during the lifetime of an individual. So before gene editing, they had tried their hands with gene replacement therapy, where they tried to replace defective genes. So it had a degree of success, some degree of success, of which we will be discussing in my next presentation, that is on 27th. I will be dealing exclusively on that. So how do we overcome off-target effect when using zinc-finger nuclease? So as I said, it is difficult to overcome such kind of zinc-finger, nucleases when it goes off the target. So how methylation protects DNA from restriction digestion? It's basically, as I told you, so methylation is a kind of a tagging which makes the restriction enzyme recognize what is me, what is self, and what is non-self, what is me and what is foreign. So that is how the methylation protects. That is how the methylation protects the DNA from restriction digestion. So thank you. With this we come to an end of today's lecture, and it was very nice interacting with you, and look forward to see you once again. Thank you very much, and have a wonderful day.